What I'm going to talk about today is an area of research I've been interested in my whole career, and that is how do we encode memories in our brain. Of course, when you learn something, it's got to be encoded physically in your brain in some way, and that's got to last for, in many cases, decades. We remember very traumatic events or your mother's kitchen. These very salient memories can last for many, many decades. And of course, we have this 3-pound squishy structure up here. So how does that brain encode those memories in a biochemical molecular way? And that's what I've been studying for the last 30 years or so. So if we look at the brain, and just very quickly, background, of course, the human brain is a network of 100 billion neurons, and it also has astrocytes, Jeff. I'm not leaving it out. But the brain has this network of billions of neurons, and these neurons are interconnected at synapses, which I'll discuss in a second. And they form unique neuronal circuits in various areas of the brain, and it's these complex circuits that underlie our complex behavior. And so one prevailing theory in how memories are encoded is that you modify the strength of interconnections between these neurons, strengthening some, making new connections, weakening others, and so you make a unique circuit in different areas of the brain that actually physically encode the memory. So when you reactivate that circuit, that's the remembrance of that memory. So there's been a lot of interest in how do you modify synaptic connectivity in the brain, how do you generate new neuronal circuits, how does experience impact these circuits. So of course these circuits occur through these synaptic connections. So two neurons here are illustrated. A neuron which has this elaborate process called dendrites here is receiving up to 10,000 synaptic connections with other neurons. And so there, in the human brain, there are quadrillions of synapses. So right now, your brain is firing away with quadrillions of synapses. And they receive this input, and then they send out an electrical signal down the axon, which then ends at a synaptic connection shown here. And so what happens at this synapse is illustrated here. This is a blow-up of the nerve terminal and the first cell, and where it's in contact with the second cell. And so the action potential comes down, the electrical signal comes down here, and converts this signal into a chemical signal. It releases neurotransmitters. And then these neurotransmitters diffuse across the synaptic cleft, the gap between the two cells, activates receptors, neurotransmitter receptors on this side, which then convert it back into an electrical signal. So this is how synaptic transmission occurs, and but as I said, this is not a static process. It's a very dynamic process. The strength of synapses are changing all the time. Hopefully, right now, some are getting stronger and weaker in your brain. And you might remember part of this talk, hopefully, tomorrow. And so you have to modify synaptic strength. And so we've been very interested in how does environment and learning alter synaptic strength and regulate circuit formation in the brain. Now, my lab and many labs across the world have focused in on the receptors, these neurotransmitter receptors, because they receive the signal, they mediate the response to the neurotransmitter. So if you can regulate how they work, or then you would dramatically affect the strength of synaptic transmission, and that could play a role in sculpting the circuits encoding memories. So this type of plasticity, what we call synaptic plasticity, the rapid and long-lasting modification of synaptic strength, has been studied for the last 30 years. And this is just an example of the classic form of this plasticity. I'm going to show you how this is defined in the area of the brain called the hippocampus. This is a brain slice of the hippocampus. And the hippocampus, many of you may know, is very famous because of a famous patient called H.M., who lost his hippocampus due to surgery to control his epilepsy back in the 1950s. And after they performed this surgery to remove the hippocampus, H.M. could never form new long-term memories. He could remember his childhood, but he could never encode new memories. So he would have to reintroduce himself to his doctors every day. So this area of the brain is really critical for long-term memory. Now, what electrophysiologists have found is very dynamic synaptic plasticity in this area of the brain. So if you stimulate one neuron and then record from another neuron and look at the strength of synaptic connections, this is, in general is very stable. So if you stimulate and record, in this slice of brain, you get the same response, one after another. However, if you give a very high frequency stimulation, one that in theoretically could be similar to a learning environment, an enriched environment, and this is a very high frequency stimulation. Within a minute, you see an increase in the synaptic strength. So this is measuring the synaptic strength. You stimulate and record, you see this, the strength of the synaptic response has gone up. Now this is very rapid, and it can last for, in this piece of tissue, several hours. But in the intact animal, it can last a year or more. So this is exactly what we as neuroscientists are looking for, something that can be alter synaptic strength very quickly, but can also be very long-lasting, something that can last the lifetime of a memory. Now, in the same area, there's a similar process. This is called long-term potentiation, because it potentiates synapses. And in the same area, if you give a different type of stimulus, you can get something called long-term depression. And this is a form where you decrease the synaptic strength. And so here you give a very low frequency stimulation, and you can get long-lasting synaptic depression. So these types of plasticity, this positive potentiation and negative depression, are thought to be what's underlying the sculpting of these circuits involved in memory. So over the last maybe 15 or 20 years, there's been a lot of work from many labs, my laboratory, and actually in collaboration with David Linden, and other laboratories all over the world, have come to a model for how these synapses are regulated during long-term potentiation. So remember, long-term potentiation is this process that strengthens synapses. And what we and others have found is that what happens during LTP is the name we call it, is that receptors are actually recruited from intracellular stores and then inserted into the plasma membrane. So these additional receptors then make the synapse stronger and increase the connectivity between those neurons within the circuit. Now the opposite occurs during LTD. So here we're going to look at LTD, long-term depression. During LTD, receptors are removed from the synapse and brought internally into the inside of the cell. And this decreases the density of receptors here, and that decreases the synaptic strength. So this, we think, are the major mechanisms for controlling long-term potentiation and long-term depression, the removal and addition of synapses, of receptors. Now, we've been studying this in many different areas of the brain and for underlying different types of memory. And we've also identified a large complex of proteins that are involved in regulating this receptor movement and recruitment to synapses. And interestingly is we've gotten interested in the pathology of this, in that many of these genes that we've discovered and others have discovered are found to be mutated in intellectual disability, autism, schizophrenia, and also involved in Alzheimer's disease. So a lot of these diseases seem to be disruption of this normal synaptic transmission and synaptic plasticity. For example, KIBRA is a gene that's a very common, has common alleles. So 50% of you have a certain sequence in your DNA encoding the KIBRA gene, and the other 50% of you have a different DNA sequence. One is a slightly smarter allele than the other. So there's the ones who have the smart allele can remember maybe 9 to 10 words in a word recall memory model, and the rest of you can only remember 5 to 7 words in a row. So we're studying how KIBRA regulates a normal variation in the human population in memory performance. And as I said, some of these other genes are involved in autism and schizophrenia. Now, I want to tell you about a couple of things, new techniques that we're using that really are revolutionizing neuroscience in general. And I think David will talk about some of these techniques as well. Now, neuroscientists love neurons. You know, we have this love affair with them. They're absolutely beautiful. And here is a neuron that's growing in a dish. You can see these yellow signals are actually the synapses. So you can see the 10,000 synapses that's impinging on this neuron that we've grown in the dish. We mostly use mouse and rats. We study in rodent systems. But as I said, we're interested in disease too, so we're starting to work with human tissue and human stem cells. So we've been studying how these receptors move around in tissue culture. But the new methods, we're now trying to look at how receptors actually move around in intact animals while they're awake and behaving. And you know, if you said we could do this to me 10 years ago, I would say you're crazy. But now we're getting to this point where we can actually image individual molecules in the intact mouse or rat brain and watch them as they, for example, get recruited to synapses during LTP or taken away from synapses during LTD. So here, what we've done is create a cranial window in a mouse brain. There's a little dental cement and a little cover slip and some very careful surgery. And you can actually see the surface of the brain shown here. Remarkably, the mice, after they recover from surgery, they run around for months and they don't even notice these little windows in their brain. But what this allows us to do is then actually put the mouse under a microscope, and this is a anesthetized mouse, under a microscope and actually look into their brain. So I won't dwell on the animal, so I can move next. So what we do is we actually generate mice that are expressing fluorescent receptors. So that we're actually, these receptors that we study are genetically encoded with what's called GFP. Many of you may have heard of this protein. It's a green fluorescent protein. And we can image the receptor using this microscope. So here we're looking at the top surface of the brain, and this is an intact live animal. And we can zoom in, and we're going to do sections and zoom in with the microscope to about 1/4 of a millimeter into the brain, the surface of the brain. And the green is the receptors, and the sort of magenta is actually the structure of the neurons. And so you can see as we go down into the brain, you're looking at these processes, the dendrites, and then down at the bottom, you start to see the cell bodies. And you can actually reconstruct this whole area of the brain in 3 dimensions using this microscope. So this really gives us incredible power because we can zoom in, and so this is our images of synapses inside this intact animal. And we're watching, in this case, we're watching receptors get recruited into synapses. So here is a synapse that, before we stimulate sensory information, sensory stimulation, has this many receptors in the synapse, and then you can see they get more and more receptors build up in the synapse, making the synapse stronger and stronger. So now we can use these techniques to actually watch this process in intact animals. In fact, we're trying to teach the mouse a trick underneath the microscope, so we can watch as it's learning the trick. What's happening to the receptors? Do they go into the synapses? Are they stabilized? And to really watch this process in real time. Now, other methods we use, of course, how do you study learning and memory in mice, or how do you model diseases in mice? There's lots of behavioral paradigms. Classic one shown here is spatial learning in the water maze. Many of you have been familiar with this. This is a tub of water, which is milky in color. There's a little platform underneath the surface that the rat or mouse can escape to if it finds it. Rats and mice generally don't like to swim, and so he's searching for almost got it there, and searching for it. Sometimes actually rats like water and they sort of float. They just sort of, so you have to throw them out. But but here he finally found the platform, And then you can see he very rapidly, within a couple days of training, finds out where that platform is. So that's one form of learning. Another form of learning we do is fear learning. And in this case, what we do is it's a fear association. So we present a tone, and then we present a light shock. Now, we're not torturing animals. We do this to humans, too. So so here, what we do is we put them in here. We present the tone, and then a slight shock, and then they respond by freezing because they're afraid. So we can measure this very accurately, as you'll see in a second. We put him in a different context and let him walk around, and then we present the cue, the tone, and he'll freeze. And you can measure the time he freezes and the percentage of time, and then when the tone goes off, he moves around. So we've been very interested in not only how you make memories, fear-associated memories or spatial learning memories, but more recently we've been interested in how do you get rid of them, how do you erase, especially fear-associated memories. And so we started studying this type of fear association, and the classical way to reverse fear associations is it's called extinction. And it's very analogous to behavioral therapy in humans, where the person has a traumatic event, you expose them to the event, you talk about it in a therapy office in a safe environment. Or actually the military now is doing virtual reality to allow people who've gone through trauma to relive the traumatic battlefield experience under safe conditions. And so this re-exposure therapy can decrease the fear association. Now in rats and mice, what you do is you just present a tone 20 times in a row, and they figure it out that they stop freezing because they figured out that it doesn't associate with the shock any longer. Now this classical extinction erasure of the fear association, it works. You can come back the next day or so and present the tone, and the mouse will not freeze. However, just like with humans, this technique doesn't really work very well. You get within a few days or weeks, you get a reversal, and the tone now induces freezing. And this is especially true if you give them a cue. For example, expose them to that chamber that they were initially shocked, or with humans if there's a loud noise or something that reminds them of the trauma. So we were interested in, is there a better way, or can we figure out a better way to get true erasure of the original fear association? And it's a little bit technically complex, so I'm going to really just present the data schematically. And what we found is that when you induce this fear learning, you recruit receptors, just like I mentioned before. You produce LTP. So you strengthen the synaptic connections, in this instance in the amygdala, which is an area that's very associated with fear. And so we think that this strengthening of synaptic connections is important for encoding that memory. So work from our lab and many laboratories across the world. Now, we were interested in this, and it turns out these synapses are very stable. These receptors are very stable. You cannot reverse this process very easily. But we started studying the real details of this type of plasticity and looked over time, and what we found is a really remarkable a choreography of receptor movement in and out of synapses. That within 24 hours after the initial fear learning, these receptors are removed, but they're replaced with another somewhat unique type of receptor that we had been studying. So this occurs 24 hours after the fear association. And then over the next 7 days, these receptors then recycle back out and are replaced by the normal receptors. Now, we thought this was somewhat funny and didn't really understand why you would want to do this. But what we found out is that these receptors, as I said, are very stable in the synapse, but these receptors are very unstable. And so during that time, that 24-hour period where the receptors are here, this potentiation is very unstable. And we could actually induce the removal of the receptors by engaging a form of LTD And this actually truly erased the memory trace. So it really, we think, erased the memory association of the fear. And this was, we think, is permanent. So by reversing this LTP phenomena, removing the receptors, we erased the memory engram. So this is interesting, but the problem is it's like a stroke. You have to treat immediately right after the fear association, which in theory you could do. But we found a way to reopen this window. So if you remind the rat or mouse of this event 2 weeks later, all of a sudden the window reappears. So in theory, you could say with humans, if you remind the person of the traumatic event a year later, this window may open, and then you could be a target for therapeutics. So the way you target this process is using drugs, and so we're now trying to develop compounds that will activate this LTD mechanism. It's called a positive allosteric modulator, and so we're doing this in rats and mice. And so what we do is we do the fear conditioning. Then the next day we just give them a single dose of this drug, and our prediction is that it will reverse the plasticity and reverse the fear association. So that's work in progress. So just in summary, we've been studying learning and memory. In the field, we know a lot more about learning and memory, and we're focusing going forward on circuits involved in encoding these forms of memory. And I can't leave out the people who do this work. This is my team of postdocs and PhD students who work on these projects. And then in this day and age, as you all know, we're all suffering from lack of funding. And so I got to acknowledge funding from Howard Hughes Medical Institute, NIH, as well as private philanthropy, the Simons Foundation, and a grant on Alzheimer's disease, actually. So I didn't talk about our recent work on Alzheimer's disease as well.